This is Popping the Bubble with hosts Sandra Ponce de Leon and Pete A. Turner.
Hello, everyone. My name is Attila Hajdu. I'm the CEO of Stelvio Oncology, and you're listening to Popping the Bubble.
All right.
That's fantastic.
Awesome. Hey, Attila. It's so great to have you here. Well, we're here at Indie Bio Accelerator, and you are one of the companies that was recently accepted into the accelerator, and you're doing amazing work that's going to save millions of lives. So thank you so much for joining us.
My pleasure for being here.
Yeah, so Stelvio Oncology. So we were just getting into what you do, but maybe if you could just give us a little bit of a brief summary here on what you are looking to accomplish.
Yeah, so Stelvio Oncology has developed a personalized approach to treating glioblastoma, which is the most lethal form of brain cancer. And there's about 200,000 people that have the disease. And over the last 40 years, there have been very little in terms of advancements made towards the standard of care for the treatments for these patients. And so that's what we're hoping to change. We're hoping to change the entire treatment landscape. Because what we do is we're not like traditional therapies where like chemotherapies and radiotherapies kill the cancer cells and the cancer always comes back. Mm-hmm. So what we do is we actually take a cancer cell and turn it into a healthier cell.
Wow. So what are the traditional treatments for glioblastoma? Sorry, how is it that you say it?
That's how you say it, yes.
Did I say it right? Oh, wow, okay.
Yeah, no. The tumor starts in the primary area of the brain. So this is why within 14 months, most patients die from their disease.
Wow.
So the treatment has to happen very quickly. And so standard of care is surgery. So brain surgeon tries to cut out the tumor, which is very star-shaped and penetrates into the brain.
What's
like
the survival
tentacles.
rate of that operation?
That is a really good question. I know that the median overall survival for patients that have had surgery and chemotherapy, temozolomide is the standard chemotherapy and radiation. The survival rate is median survival rate is 14 months. So
even with all that.
With all of that.
Wow.
Yes, and so that's what we're hoping to change. And so, and we are cautiously optimistic that we have discovered two small molecules using our technology that turn cancer cells into healthier cells.
How did you get into this specific? I mean, this is a very specific kind of cancer. How did you latch onto this?
It's a really good question. So I have over 20 years of experience in Big Pharma with GSK and various senior management positions. And GSK is a multinational global pharmaceutical company with about 150,000 employees all over the world. And so that's my background, and I am passionate about oncology, and I think anyone that is in this space has to be passionate about what they do, and that's what drives them, and I'm definitely one of those people. And so throughout my 20-year career at Big Pharma and then a small biotech where I closed a $290 million deal, licensing deal, I obviously have developed a broad network of physicians, within the communities, volunteering, doing various different activities, and so cancer was always sort of a passion, and so I met and helped raise capital for many physicians that are principal investigators of clinical trials. And they introduced me to other physicians, and then that network grew. And then all of a sudden, these physicians are doing amazing technologies right in the academic institutions and the research institutions. And that's what led me to David Spanner, and then who's an MD PhD at Sunnybrook Health Sciences Center in Toronto. Then I met David Andrews, who's head of biochemistry at Sunnybrook Health Sciences as well. And they introduced me to Alexei Turskic, who is now my chief scientific officer, who was a co-inventor of the technology which came out of Sanford Burnham Medical Discovery Institute, which is in La Jolla, near San Diego. So we have, yes, we're a small company, but we have a very large footprint because our lab is where the $20 million machine is, because we're very deeply rooted in machine learning. And so the technology is using phenotypic screening using epigenetic marks, and which is where machine learning comes in, because it's a very complex technology. So we're in one of the hottest areas of biotech right now, is combining biology and machine learning. Huh. And so the $20 million machines in San Diego, and then David's the guru of David Andrews is the guru of machine learning, where the programming and all the analytics occur. And then obviously we're at IndieBio here in San Francisco.
Yeah.
So you said a couple of big words there.
Really big words.
Yeah. Phenome.
That's good.
Yes. And so go back to that. And also you don't just get to say, yeah, we take the cells and we turn them over to good cells. I mean, so how are you guys, what's the process? Are you looking at microRNA and taking it and reprogramming the code, or what's going on? What enables, without revealing your big secret?
That is a $100 million question.
Thank you.
That is the $100 million question. You know, if we knew the answer to that, it would be, and the investors are asking us the same question. The investors are saying, what is the mechanism that is turning cancer cells into normal cells? So that's the $100 million or billion dollar question right there. And that's a bit more complex to answer. If we give that away, then obviously there goes our IP.
But it's all related to a specific molecule that you've discovered.
It's all related to the small molecules we've discovered. So what we're doing is how that cancer cell turns into a normal cell, or a healthier cell, sorry. We can do this in the lab, right? Uh-huh. So we can, in tissue culture, we can take a cancer cell and through tissue culture techniques turn it into a healthy cell. Mm-hmm. That's not a very useful to a patient who has the tumor inside of their skull.
Right. But has that been done before? Is that something new that you guys are doing?
It's been done before in the culture. Yeah, it's been done before. Yes, absolutely. And that's where this evolved into what we're doing.
Right.
Right? Because, we can do it in the lab in the tissue culture, but we need to do this as a treatment.
Right, exactly.
That works. So we're excited because we've discovered 2 molecules that do this in vitro, so we're further characterizing these molecules, and then obviously the next step is looking at in vivo efficacy. So what does this do in animal models? So we have what's called a xenograft mouse model, which is it's sort of a chimera between a human and a mouse. Mm-hmm. So you take a human cancer and give it to the mouse, and the mouse is living with this cancer. So we've got these xenograft glioblastoma mouse models, and then we're obviously going to treat the mice with our drugs that we found.
Right.
And the primary endpoint will be slowing of tumor growth.
Right.
And we need to show this statistically so that science accepts only valid statistics and results. And so we need to do this, and we'll have these results within 2 to 6 months, depending on how things go. So if we can slow tumor growth in mice, then, that gives us more insight into the $100 million or billion dollar question.
Right. Okay. So you're going to slow the tumor growth in mice. How invasive is the treatment? I mean, we're still talking brain surgery and getting.
So this is a very interesting question too. So how we're going to dose the mice is subcutaneous. That means under the skin.
And then from there, is it a repeated treatment that you do then, or is it just a one-time shot?
Yes, so that's exactly what we need to understand as well. Is this a 14-day course of therapy, once a day, twice a day? So what we know is the drugs we have discovered are working in the nanomolar level. So what that means is it's exponentially below the current treatment dosage amounts. So we can use very small doses to have a bigger effect. And so, and then of course how we figure out the dose is dose escalation. And there's a protocol for that, and basically you escalate the dose until you see something's happening, and then obviously you have a range. I mean, these are very standard approaches to dose discovery, but dose escalation is the main way.
And isn't there a maintenance dose after that you have to do to keep the cancer from reoccurring, or are you done once you've done the therapy?
You know about treatment, don't you? Because you're talking about maintenance doses and induction.
I just know some things.
You know about this. So in this particular case, we don't know, as I'm sure you're aware, and from hearing you talking, and you are. So some cancer treatments do have maintenance doses. They have an induction dose, and then they have like Rituxan. You get an induction dose depending on what it's for, and then you have a daily dose of 500 per day, and then you have a follow-up dose. But with ours, I think what we'll see is we'll see treatment between 2 and 4 weeks, and then you stop the treatment. So you take what we're trying to do is you take three different animal groups. So you have standard of care controls, right? So it's TMZ plus radiation. Can't really do the surgery.
What's TMZ?
Is telozolomide. It's the chemotherapy.
Okay,
so
got
we
it.
take one arm will be standard of care, so TMZ plus radiation versus TMZ plus our drug, right? Versus our drug alone.
Got it.
We always have to have comparison to standard of care. How do you stack up against standard of care, right? Everyone wants to know that. And so that's how we have to. And so what you do is you have those three arms. You do the treatments for 2 to 4 weeks, and then you stop the treatment, and then you observe. So you do tests, blood tests, and at the end of the study, we'll obviously stop the study, and then we'll do morphological assessment as well. So that's the plan.
Yeah. Well, I mean, it sounds like, from what I've heard about drug approvals and the FDA, I've always heard it to be a very long, drawn-out process, like something like 10 years before these drugs can actually be in market once the testing starts. So is that the schedule that you're on? If you do have a breakthrough, is it going to be another 10 years before the drugs are available to people, or is there an acceleration schedule, or how quickly are we going to be able to see the benefits of your work?
So, Sandra, that was a really good question, and for many reasons, because the drugs that we've discovered are actually FDA-approved drugs.
Oh, okay.
Okay, so we know
they're already approved.
Yeah, so they're already approved
use
because
for other
exactly.
cancers? Oh, I see. Okay.
So this is where the very interesting question comes in, is that they don't have any commercial value to anyone really, other than the company that has the FDA approval, but the FDA approval is likely for another indication. But we know what we're seeing now is that these drugs turn cancer cells into normal cells.
Are they drugs for cancer, or are they drugs for other ailments?
They could be drugs for other ailments, but primarily drugs for cancer.
Okay.
So this is the interesting thing. We have a panel of 1,200 FDA-approved drugs for which we screened and found 2 hits. Oh. But imagine the opportunity when you screen a library of 10 billion compounds, not 1,200. And so the short answer to your question is, what happens if we have discovered that these 2 FDA-approved drugs turn cancer cells into normal or healthier cells? What do we do then? Do we try to license another compound in a similar class, which we own the IP for? And then it's, the 10-year process, as you said, and $1 billion. Each of these is $1 billion bet, by the way, right? Every single drug that you're trying to get to market is $1 billion bet.
Wow.
And so that's where we have an advantage, because our approach is personalized. It's targeted therapy.
Right.
And so the reason it's personalized is because every tumor is a person. And that's where our starting material is from. So we take biopsies right from the patient. And that's what we use as a starting. So it's patient-derived samples that we're starting from. And so the idea behind personalized medicine and precision medicine is that traditionally, if you give chemotherapy to patients, there would be a lot of patients that would not respond and therefore had the toxicity. So that's not very useful in moving forward. Why have unnecessary toxicity with no efficacy? doesn't make sense. So if you know, for example, which drugs patients will respond to, wouldn't that be a better future?
Absolutely.
So this is where precision medicine and the way that I phrase the question is, what if we could turn a cancer cell into a healthier cell without the unnecessary toxicities of standard of care, chemotherapy, radiotherapy, and the fact that we're not trying to kill the cells, we're trying to...
Or remove them.
Yeah, exactly, or remove them. So that's the idea. So precision medicine, I mean, it's better for the patient. Probably one of the best examples is TiCurb, which is I'm very familiar with because I worked with that product when I was at GSK. TiCurb is for...
I'm sorry, what was that?
It's called TiCurb.
TiCurb, okay.
TiCurb, it's basically for metastatic breast cancer. Mm. So the interesting thing about this is, because it's one of the best examples, is that there's a protein called EGF, and if it's overexpressed, then patients will respond to a drug called Herceptin or HER2. If HER2 is overexpressed, then patients will respond to Roche's drug and our GSK's drug, TiCurb. So we know if every woman in the United States has access to Oncotype DX, which is this test, which tells you if you overexpress HER2, and therefore you would be a candidate for TiCurb or Herceptin. So in Canada, for example, the test is $3,000, but that's what we have. And I'm using the analogy because it's, as I said, we have the personalized approach. We're starting off with the tumor. Mm-hmm. So that is a built-in diagnostic.
Right. When you say personalized, and then you also talk about precision medicine, are those one and the same, or
yes,
is there a difference?
Yes.
Okay.
Yeah, they're the same. They're used interchangeably, so it's the same thing.
And is that relatively a new way of treating patients, or?
It's I think ever since I think Herceptin's one of the few, and the reality is most drug companies don't build that into their clinical development programs. And unfortunately, another one is melanoma, right? So again, Roche, and probably the best example is Gleevec from Novartis. So that was a total game changer. And
what's that used to treat?
It's colorectal cancer.
Okay.
Yes. And so, there's drugs like for melanoma, which if you have a specific mutation, the diagnostic test can pick it up and know that you will respond. Mm-hmm. So this is definitely the way forward. I mean, this is the future.
Okay.
Versus blasting patients with chemotherapy and no response and lots of toxicity.
Right, absolutely.
It has to be precision medicine, and that has to be the way forward.
So when you're in the lab and you guys are doing machine learning and you're examining all of these billions of options and everything,
yes.
That means you're in the lab. Accidents, good ones, happen in the lab when you're doing things like that.
Yeah.
What kind of things do you foresee as being possibilities? Are there maybe not other cancer things, but maybe there's something that cures ALS or something else? Is that a possibility with what you guys do, that there could be these happy things?
Yes. Now you're onto it. Now you're onto something big. So the truth is that our technology is agnostic to tumor type. So we are looking at this now in breast cancer to build a pipeline. We're looking at this in all cancers.
Wow.
And so it's not just glioblastoma, but breast cancer, colorectal, prostate, leukemia, you name it. And the other angle to your question is, do you have any positive benefits to your drug? And for example, there's an antidepressant called Wellbutrin, which is SSRI, and it was seen that the patients that were taking this drug for their depression actually stopped smoking. So the exact same drug was repackaged and relabeled as something called Zyban, which is for smoking cessation.
Wow.
Yeah, and isn't that neat? I mean,
That's awesome.
These kind of things happen all the time, but I mean,
Right.
you have to obviously connect the dots and make an observation, right? You have to, have patients in under your care, and all of a sudden, you're smoking a lot less.
You see how big my biceps are?
You've been working out?
Yes. From colorectal cancer, I did like bodybuilder.
Yeah, exactly. Yeah, there you go.
So when you guys have this program and you're working on animal testing and those kind of things, how far out to being able to do like human testing and that kind of thing? Are we talking, the drugs are largely approved?
So it's after we have our, the stage we're in now is the preclinical stage, right? Mm-hmm. So if we show in vivo efficacy, then we'll move into, pharmacokinetics, toxicity, those kind of things, and then that will take, let's say, a year, and then 9 to 12 months, and then I think what will going back to Sandra's question, what will accelerate that is partnerships, and right now we're talking to some pharma companies, and obviously within my network, Genentech, for example, because you're here, and J&J. I mean, partnerships. I mean, the list of things to do to get it to market is immense, and we don't have that resource.
Yeah.
So what accelerates this is, and what I can see as the future is partnering with Big Pharma, who has the resources and can dedicate staff. Resource to me is people, money, time. That's basically it.
Yeah.
And so then the partnerships will accelerate the growth. We'll obviously, we can do this. We can screen their libraries. Big Pharma have compound libraries of 10 billion.
Right. So this is where the machine learning comes in,
because they can't do it.
Right. And is that something that's unique to
us, to
what you guys are doing? Is applying that machine learning approach to
this? Exactly.
Yes.
It's unique to us, and that's our sweet spot. Mm-hmm. Is because we have the ability to use phenotypic screening with epigenetic marks, which incorporates the machine learning. So, and then we're the only ones in the world doing this. And so the interesting thing there is... Sorry,
what's an epigenetic mark?
So epigenetic... remember we talked about painting the nucleus?
Right.
That's what it is.
Okay, this is... Okay.
So painting the nucleus.
Let's describe that.
So, okay. So inside a nucleus, there is chromatin, which is part of DNA. So DNA is made up of sugar base phosphate, which is DNA. Mm-hmm. And then there's also chromatin, which is the protein that links strands of DNA into chromosomes.
Okay.
Right?
Yeah.
So you think of the nucleus. So there's about 50 epigenetic marks in the nucleus. Mm-hmm. And it's all based on open and closed chromatin. That's all there is. So if you have open chromatin that has a histone methylation site or acetylation site, we can label it with basically immunostaining, standard immunostaining. So basically what we're doing is labeling open and closed chromatin, mm-hmm. and basically painting the nucleus.
Right.
Right? Mm-hmm. And there's about 50 marks. Mm. And then what we do is the information that we have is quantified. The spatial information is digitized, hence the digital pathology, and then it's projected into a multi-dimensional space of about 500 dimensions, and this is where the machine learning comes in, mm-hmm. right?
Right.
But we're taking that 500 dimensions. All you need is 2, right? And then moving into 3D now, and live cells. Uh-huh. So that's where the machine learning comes in, because a human just can't do that, right? So because there's so many different marks.
Right. Well, it sounds like that approach actually has a potential to disrupt the whole process of drug discovery and this kind of therapy.
We're toppling industries here, and that's what we're doing. I mean,
wow.
We're completely. That's our hope, anyway. Yes, for sure.
You're talking about a lot of high-end, big brain things.
Yes, so, I'm trying to take it down. I'm trying to do my best.
This is great.
I'm trying to do my best.
What does Attila do on a day-to-day basis? I mean, are you putting a lab coat on and squirting things into tubes? Are you?
I could.
Are you calling people on the phone?
Or dishes.
Right.
I could. Well, I'm designing studies. I'm talking with our investors. I'm pitching people. I'm a father and a husband. I talk to my kids who are back home in Vancouver, and make sure that they're all looked after. So I do a lot of things. I mean, but yeah, of course, the priority right now is obviously is to prove the science. I mean, we're a scientifically driven company, so I mean the science is so I'm working to design the studies with our CSO, and so moving that forward, really overseeing everything. I mean, as the CEO, my job is not really to actually do the science. It's to sort of set the vision and get out of the way. Right.
You're definitely a science-driven company, but are you a computer science-driven company with your machine learning that uses
that's a part of it,
medical science platform?
But no, we're a drug discovery company.
Okay.
And so the machine learning is a tool, just like a craftsman has a hammer and a nail for their tool. Those are just tools.
Yeah,
right.
Right.
So
I was going to go with
woodworker,
like plasma cutter, or 3D printer, or something.
Ghostbusters. Yeah. Yeah. No. So that, I mean, so basically, I connect the dots. That's really what I do. I mean, I connect the dots between where the technology is, find a piece of IP, or do we need to license this, or raise capital. So right now, I mean, I'm here competing against other companies, and these are all great companies. So, but I need to essentially tell the story so that it attracts the ear of someone who can take a checkbook out and write me a $1 million check. Mm-hmm. So that's
just $1 million. Is that all?
Well, when I arrived here, I said I'd like to raise $100 million, and then they all looked at me like I was, on something. I said, really, I do want to raise $100 million, right? So right now we're working on a deal for $5 million, with an upfront of $1 million and $4 million in milestones based on the two milestones we discussed about the in vivo efficacy and the confirmation with gene expression. So, I mean, that's what I do. And then the other thing I do is like basically we work with lawyers. I mean, we're working with IP lawyers to patent all this stuff. I mean, there's about 35 different claims in our patents, right? And so, I'm sort of leading the social media. You saw the Facebook page. Well, somebody has to sort of think of these things, right? Mm-hmm. And do them. And so I wear a lot of different hats. Obviously, I have a CFO hat on. I have a, sign, if people need me to pipette things, sure, no problem. I've got a treasury hat on.
Yeah, you do it all. Well, I mean, in a startup, you are wearing, you're wearing a lot of different hats.
You have to juggle many different balls. Mm-hmm. But, at the end of the day, it's right now, it's the science and then the funding. I mean, for the science. So that's what we're focusing on. Right.
And so ultimately, when you're developing, just from a marketing perspective, I'm curious, who's your customer that you're speaking to on your website? I mean, are you talking to pharma companies?
Well, you are. That's where I think this is really, that's a really interesting question. So the customer is very important. So it's multi, but I think the most important customer is pharma. So we're talking to pharma companies, Genentech, J&J, all of the big ones. So we have obviously a target list of companies that we're focusing on, Novartis, these companies that are in the oncology space. And then, of course, the customer is also the investor.
The investor, yeah.
Right? Because definitely, and the customer is also the patient.
Right.
You can't forget about the patient.
Of course not.
Because, that's why we're doing this, right? Mm-hmm. It's focusing on the patient. And, the customer is the community and the families, because I've had, in my family, cancer has impacted my family. And it's a tremendous burden on families, and so the customers are the families,
right? And
so the financial institutions. So it's society, really, and if we're toppling industries, it's multiple. Mm-hmm. Mm-hmm. But that's so your marketing question is a good one, because that's about getting the story out and creating a movement, and so marketing is critical because the story has to be heard. I mean, I'd like that Facebook page to have 1 million likes, not 10 or whatever it is now.
Right. Well, it's creating the groundswell.
And now it's 11.
Yay! Thank you. High five.
We'll do our best to get your Facebook likes up.
Thanks. But it's telling the story, and it's a really important. It's critical.
Absolutely.
So I had GSK's marketing, for example. I mean, it's a marketing machine, right? It's what Big Pharma does really, really well, right? But they also have billions of dollars of cash in their war chest.
A lot of money. Right. The name Stelvio, which is the name of the company, what does that come from? And then why not Stelvi, like singular?
Yeah, so Stelvio, if we ever have a drink together, I'll show you some pictures of me. But basically, the Stelvio Pass is an iconic pass in the Italian Dolomites. It's tops out at about 2,800 meters. So, I mean, what's the biggest mountain around here? Probably Lake Tahoe. So it's a lot higher in Lake Tahoe.
Yeah.
So I did this race called the Haute Route. It's part of it's kind of a mini Tour de France or Giro d'Italia, whatever you want to say, over the Italian Dolomites, Swiss Alps. And it's all about, imagine climbing 20 different mountain passes that are literally touching the sky over 7 days. I mean, to me that it's all about conquering cancer, and you can't just show up one day and decide you're going to do that. And that's what cancer patients have to face too. It's like they have this huge mountain to climb. And so the vision there was, we need to do this together, right? We need to do this together as biotechs, as pharma companies, as investors, as patients, as families, because you can't do it on your own. So Stelvio is sort of imagery for overcoming cancer together, or overcoming mechanisms of resistance together. So that's really where it comes from. And basically, we're climbing the mountain together, right? So that's the idea.
And will you hire Dolomite as a spokesperson for you?
Could. You need a dog named Dolomite. Little mascot. Yeah, that'd be cool. Yeah. So that's where it comes from. Yeah.
Obviously, if this works, and you're pretty positive about it because you're betting.
We're cautiously optimistic is the words that we've chosen.
Well, I'll be positively optimistic for you.
Thank you.
Are we talking about a cure for cancer in our lifetime then?
Absolutely. I think even if you follow the 10-year sort of average, right? I mean, that's, I think the three of us sitting here will live definitely for 10 more years, for sure. So that's what we're doing is we're curing incurable cancers right now.
Amazing.
Yeah.
Wow.
Yeah.
Yeah, you let that sink in. I mean, that wasn't possible. I mean, this is like Wright Brothers flying type stuff.
It is.
No one was able to fly.
You're at the forefront. Absolutely.
Someone figured it out.
Yeah.
And it's incredible.
That's just it. And I think Einstein once said it's impossible until it's done. Same thing, right? So.
Is it easy?
No. I mean, nothing worth having is easy. And that's a big life lesson I think that I've learned. I mean, if it was easy, everyone would be doing it. And that's the thing. That's why you challenge yourself and others to take on these big problems because, and there's high risk. And the people that are in the high risk jobs are there because they can overcome that risk.
Mm-hmm. Sometimes, though, something is easy once you've seen it done, and
Sure.
it's about perspective. There's that story about Christopher Columbus and everybody's saying it's no big deal what you did.
Yeah.
And he's like, can you make an egg stand on end? And everybody's like, you can't do that. You took it, went and broke the bottom of the egg and it stood up.
It stood up.
As soon as you see it done, it's easy.
It's easy. That's what I mean. Yeah, exactly.
Yeah.
Yeah, and that's the point is that if everyone's doing it, then it's easy, right? If no one's doing it, then it's hard. So right now we're at the point where no one's doing it, and therefore there's high risk, but there's also high reward, right? Yeah. And so, but that's why we're doing it is because it is hard, and so you hire the smartest people, you hire the people that are passionate. I mean, it comes down to the people behind it and how you de-risk it moving forward, right? And so, I think, I mean, there's some CEOs out there that you hear them saying, well, I'm usually not the smartest person in the room because if I am the smartest person in the room, then there's a problem, right? So they hire super smart people and they're able to connect the dots and say, okay, so this is what you do well, this is what you do well, and now we just take it all together, bring it all together, right? So,
what are some of the challenges that you faced along the way in this this part of your entrepreneurial journey?
Financing.
That's the big one.
Yeah, I think so because the challenges are, how many VCs will we have to pitch before we actually get one to write a check? 400, I don't know. So one of the benefits of my previous role with a biotech company was I made a network within the investment community. And so we have a group of investors that I know well and I've tapped into, and one of them has committed for the $5 million. So great.
Yeah.
So we just need 20 people like him.
Yes.
And then there's our 100 million, right?
Yeah.
So that's all we have to do. So, but it's at the end of the day, I mean, I think the people that you know really well that have a lot of money, they're investing more in the people, right? They're not really technological. They don't have any idea. They're just, okay, Attila, you've done this before and we know you can do it. We're going to give you some money, and then, hopefully, you'll give us 25% of your company, and then, Big Pharma will come along and write you a bigger check.
Is the environment different this time around than the last time that you went about it?
It is, because as the CEO, I'm now an entrepreneur, and so what I'm learning now is, I'm being reprogrammed from being a corporate type person, which is where I came from, and the culture there is quite different than here. And so I'm being reprogrammed to be an entrepreneur and be, NDBio is doing a tremendous job there in doing that and helping people become entrepreneurs. Because, before I used to say, okay, let's do this study, and that will cost $100 million, that will cost $100 million, that will cost $100 million. Push a button, the money goes in the account, boom, it's done, right? But I have to go find that $100 million.
A little different this time around, yeah.
yeah, because as I said, it's like Big Pharma has lots of resource, so it's like, you want a round TV, let's go make a round TV. So. Right. But
yeah.
So it's different. Then, that's basically it.
So as an entrepreneur, obviously you've got plates to balance. You got things spinning everywhere. And are you still learning how to keep certain, you have to do marketing, you have to do storytelling and sales and
all
on
that.
Top of the science.
Right.
Are you learning those things here as well? About how to balance out your approach to, everything needs your effort, but not everything at one time.
Yeah. So that's actually a really brilliant question, because the way I look at it is very simple. It's effort impact, right? How much effort is activity X going to take, and what is the impact? So right now, obviously, I have to go and prioritize and say, what's the least amount of effort for the most amount of impact?
Yes. And then as you do this, so the corporate you, was that person more of a risk taker than the entrepreneurial you? Like, do you what, I can't get these things wrong because you're the boss now. You've got to go find the money.
Yes, exactly. So I understand.
Is there how is that evolution of?
Or is it the other way around?
Yeah, right.
Well, in big corporations, the risk taking was different because, for example, GSK was always about risk mitigation because they had a lot. Therefore, they didn't want to lose any of that. And they wanted to return for the shareholders. At the end of the day, it's building value for the shareholders. That's it, right? And the companies that do that well survive. And the companies that don't do that well don't. So that's the model. You get rewarded for innovation. And the pharma landscape's completely changed because many years ago, it used to be the pharma companies would in-house discover compounds, but that's not the case anymore. They're, if you say there's 1 million PhD scientists doing the kind of work that we do, and Big Pharma owns 10,000 of them, where are the other 99%? Right?
Mm-hmm.
Yeah.
They're out there.
Right.
That's us.
Yeah.
Academic institutions, biotechs. So that 99% is out there. And that's what's happening with Big Pharma are really just making bets. So the risk there, what is the risk? It's like gambling. You go, if I make 10 bets, one of them is going to, come up big.
Right.
They're more like acting like VCs now and
Every
looking
pharma
for innovation through startup acquisition.
So these bets are also like
exactly
a tenth of what the cost of a study would be, like, $1 million rounding errors really for them.
That's what I used to call it, rounding errors.
Mm-hmm.
Yeah.
Versus us, we can't afford to make $1 billion bets because if we just can't afford it. We can't afford that error rate.
Right.
So right now, the failure rate in Big Pharma is 90%. So it's like, you got to be kidding me.
Wow.
So if you spend $1 billion, you're basically flushing 900 million down the toilet.
Yeah.
Wow.
Like, seriously?
Yeah.
I can't do that. You know, if you give me $1 million, then I have to make sure that $1 million is $1 million or more.
Right.
Right. So it's the other way around, is that, you know, obviously we're taking the risk, we're managing the risk with hiring the best people and the best advisors and overcoming the risk. That's what we're doing. And versus throwing $1 billion bets around.
What's the funnest part?
The funnest part is I guess seeing my kids laughing and smiling and hearing them laughing. I guess. I mean, that's the funnest part. I mean, for me, that's why I do it, I guess. But at the end of the day, in terms of the business, what's the funnest part is seeing positive results. Like, no one, I don't think, as a scientist, no one likes anything more than seeing data. When I saw the data, I couldn't believe it. I said, "Hey, Alexi, like, you know, we gotta present some data here." And I saw the you know, the dots on the chart, and I went, "Really? Like, those two drugs cause cancer cells to turn into healthy cells?" And that's the fun part, right? When you see that,
Right.
and then you go, "Well, wow, you know, we've just made a huge discovery. Like, we've just split the atom, right?"
Yeah.
So it's kind of like that, right?
Yeah.
And then no one else has seen it before. So you're the first person on the planet to see it, right?
Wow.
So it's like the first person to be on Mars or the first person to be on the moon or something, right? That's kind of fun. That'd be
Absolutely.
kind of fun.
Yeah.
So that's the funnest part from a business point of view. But yeah.
Yeah, well, I definitely can hear the passion, and it must, I mean, just the fact that you're working on something that's going to be such a game changer and save so many lives must just feel
Absolutely.
so wonderful
Yeah, it feels
inside.
awesome.
Yeah,
Absolutely. It feels great. Yeah, absolutely. Yeah, it feels awesome. I mean, you know, that's what brings me back to why I do it in the first place. It's just that passion for oncology, and you know, my parents both died of cancer. My wife's parents both died of cancer. So, you know, my wife was just 11 years old when her mom died of ovarian cancer. So I want to do something about it. I want to change ovarian cancer. I want to topple those things. I mean, imagine being an 11-year-old girl and you don't have a mother anymore, right?